So my whole interview was like completely blown out.
Oh, wow.
Okay, guys.
Great.
Oh, that's really weird. Like, if you, so many of you.
My grandparents used to have this mirror, one of those mirrors that went like this. When I was a kid, you could like turn it and you could see. It was like just kept going into infinity.
That's you.
Yeah. That's when I went down the rabbit hole, I think. Yeah. So this is the last time we're going to meet. No, tomorrow for the lab practical. The lab practical. Keep up with the Latin. I hear you guys in here. That was nice. You guys pretending like you're all bad students. It's really cute. You know, like, I don't even care anymore. I'm gonna fail everything.
Sounds like an attack.
My exact words that probably said once.
Yeah, no. How many times have we been here? How many times have you felt like this? And how many times did it work out? It's all gonna be okay. You just get, yeah, I'm telling you, I promise you. I'm here. I'm here for you. Yeah, I'll take you back to last semester. You probably, damn it, I always pull this thing off of here. You always feel like this, and it always works out. It's all gonna be okay. At some point when you're old like me, you just like, well, it's just gonna work out. It's fine. It's no big deal. Right? It'll all be over soon.
Right.
And I'll just leave it up to you what that means. But seriously, it really has been a good semester. I hear a lot of my colleagues complaining about it's a ghost town over in Lawson, Tarpley.
Yeah. They
Like,
all finish their classes a month.
I don't know. I mean, and people saying like, oh, it's so terrible. Everyone's in such a foul mood. But I feel like in Dobbs, while I'm over here, I feel like it's almost business as usual. I see my colleagues. We're here. We're doing what we can. And you guys have been here too. I really appreciate that. I didn't want to sit at home. I think like it's worrisome anyways, this whole thing. And so to have something to put my energy to, come in here, talk about animals, it's been a good thing for me. So I appreciate you guys, your efforts. And, you know, I think back to some of the labs when we were outside, that was a good distraction for me. So, you know, just finish strong, do your best. That's all anybody can ask. As for the posters, did you guys see the materials I put up? There's a rubric, and there's a couple of example posters. So if you go to that module, I think it's the projects module, and then there's an assignment for the poster. If you go in there, there are links to posters and.
I tried to access that on my phone, and it kept saying it was unavailable. So.
Okay, check. Yeah, and anybody else too. Just let me know if you're having troubles with it, because I need to get that. I'll get it straightened out. If you feel like the data part of I think the posters, there's two main parts. One is your data. Well, it all starts with a question, of course. But we've already established that. Then there's going to be data, and then there's going to be literature, right, about that question. You're not the first one to look in. I didn't see any questions come out of this class that it's a totally novel question, right? These are things that have been researched before, but you can collect some data towards answering that question. But if you feel like you're a little bit weak on the data side, try to beef it up on the literature side, right? If you feel like you maybe got a late start or you couldn't figure out quite how to get the data in, give me some, you know, in your discussion section, beef that up and talk about the next time. Sort of like, what would you do differently, okay? Don't just give up hope and say, well, I don't have any data, so I'm just going to put nothing on my poster. All right, do your best at getting closer to an answer to that question. Okay.
It does say, like, if you click on it on the canvas, it says unauthorized.
Okay, for the entire module or the?
Yeah, for the rubric, not the module, like, if you can click on the module, but like for the rubric, if you click on it, it says unauthorized.
Okay, how
and
about the posters? Can you see those example ones?
they say unauthorized too.
Okay,
It might just be something with like the links. So you could, like,
okay, as soon as, I will do that as soon as I leave here, I'll go figure out what's going on with that. But are you guys authorized? Can you show me some ID or anything? I mean, how can I be sure that you're authorized? So, I mean, there is that too. It's partly a little bit your fault too. You didn't really, but seriously, I'll get that figured out. What else? What else? Everybody's cool?
Wait, since I switched my thing from catfish to graphic, do I need to have like a different annotated bibliography?
Well, no, but, obviously change give me some literature on your poster that has to do with crappie. Now some of the literature may be overlap because again, if you're looking at a topic, you can find literature that's generally applicable, maybe about diet and about prey size or diet diversity of diet or whatever. That will maybe still be applicable, but any literature that was specific to catfish, you'll want to find stuff that's specific to crappie.
Okay.
Right. And it's the same. What do you need literature for? Your intro, what has known about the topic, why the topic is important. You might want some literature in your methods. Some people do, some people don't, but especially in your discussion. Right, for anything that you found, you want to put it in some kind of context. Right, why would does be interested in doe urine? Why, what is known about fox rolls in North Georgia? Is there anything out there? If maybe not in North Georgia, maybe in North Carolina in the mountains. So, just do your best. I think at this level of class, it's not really that it's more difficult or more time consuming, but you guys can now start thinking on your own, and I'll not be particularly giving you the literature to plug in. At lower levels, I say, here's a paper, and then where does it go in the lab report? And you guys kind of plug it in. Now you guys should be at the point where you're starting to be able to find literature and know where it applies, right? So yeah, well, okay, I will take care of the that module, make sure you guys have access to the rubric and all that stuff. And we'll just keep going forward. So these are the last few questions on this exam 3 study guide. Okay. So again, I've been using this other textbook for this part, and I think I might change the textbooks for next time. This is a really good textbook, but it's all about comparative vertebrate anatomy. It's about looking at how these things change as we go up a phylogeny. Okay, so, and as we can see, when you go from something like a generalized reptile, so some kind of squamate or lepidopteran, right, we have 2 atria, right? The ventricle tends to mix the blood, okay? But then we go, on one hand, we go up the synapsid lineage to the mammals, or we go up the archosaur lineage to the birds, and in both of those places, we get to complete ventricular separation. 2 atria, 2 ventricles with a complete separation in between, okay? So the question 13 on this study guide is, explain what seems to be the major reason for ventricular separation in mammals and in some archosaurs, crocodiles and birds. So why is this? Right? Why did evolution and natural selection lead to this complete ventricular separation? And here's the interesting thing, because it happened twice in these 2 derived lineages, it really suggests it's beneficial. It's adaptive. Independently, these 2 lineages have progressed to this design. So what's so good about complete ventricular separation? Why is that more adaptive than having blood mixed here? Okay, and just kind of step back. What's coming into the atrium? What comes into the right atrium? What kind of blood? Deoxygenated. Okay, where's it come from? It's coming from the body. It's coming from all those cells that have used up the oxygen, used up the glucose. It's coming back here. Then where's it go? Down to the right ventricle. From the right ventricle, because it's deoxygenated, where are we sending it? To the lungs to get oxygenated. Back to the left atrium, left ventricle, out to the body. Okay. In lepidosaurs and the turtles, when it gets to this point, it's not a complete division here. Okay. So why is the complete division beneficial or adaptive? What do you think?
I guess because it's not already mixed, and so like you can have a set where deoxygenated blood is like supposed to be, and then where oxygenated blood is supposed to be, so it's not all mixed together.
Okay, because what happens if you mix this and this? Then the blood that's going out to the rest of the body is going to have, what's the oxygen level of this compared to a mixed blood? It's going to be higher, right? So that seems like the obvious answer is that when you don't mix it, then there's more oxygen going out to the head. There's more oxygen going out to the organs and to the muscles, right? But it's actually a little bit more complex than this, because as organisms got bigger and they became more active, remember we had that whole discussion about being big and active. You need more and more and more oxygen, okay? So if you're bigger, say a giraffe, what would you estimate about the blood pressure of a giraffe's blood?
Pretty high.
It's going to be pretty high. Why?
They got pumped all the way up that neck, the head.
It's going to be pumped a long ways. Overcoming gravity, the pressure of gravity pushing down has to be overcome by the pressure of the heart pushing up. So blood pressure goes way up, okay? Blood pressure is high. Now, high blood pressure here, that's going to be a good thing, right? Because you're pushing it all the way up to the head. You're pushing it all the way back to the hind legs in the elephant or something. But what do you suppose about really high blood pressure here? What's that going to do? In the right ventricle. Is it necessarily going to be really good to have a real super high blood pressure going to the lungs? What do you suppose is going to happen if you're pushing the blood this short distance to the lungs as hard as you're pushing the blood all the way this long distance to the tail?
I don't feel like you're gonna get as much blood oxygenated.
It's probably, and what's going on when the blood gets here? It's going to be huge think of your alveoli, and this is what COVID has reinforced for us, is that your lungs are very gentle. Your lungs are full of capillaries. Any kind of clotting that happens in your lungs is really bad, right? Why? Because it's very porous, and if there's too much high blood pressure pushing to the lungs, it's going to force all that plasma out of the lungs, right? You can't have a high blood pressure loop going to the lungs, okay? Because it will push it right through the alveoli. Okay, so really, one of the biggest reasons for this division right here is that we want a low pressure system going to the lungs and a high pressure system going up the systemic and necrotic loops. Okay, and that's exactly what happens. This ventricle is independent of this ventricle. The amount of contraction, the amount of pressure generated by this ventricle is separate and independent from. Now, if this is one big ventricle and it's mixing and it's sending the blood out from one ventricle, you can't have two different pressures. Okay, And that seems to be the biggest reason for this complete ventricular separation. Okay. That was kind of surprising. I never thought about that before. It was a bit surprising for me to read it. Now, it makes perfect sense when you understand it. Okay. So, amphibian, 2 atria, 1 ventricle. More or less total mixing of the blood, right? We, from the systemic to the right atrium, from the right atrium to the ventricle, from the ventricle, both to the lungs, a pulmonary loop, and to a systemic loop to the body. Right, from the lungs back to the left atrium. Right, so here we have one contraction of this one ventricle sends blood out. The same pressure to both the lungs and the body. Okay. Over here, mammals, birds, because of this separation, we have a low pressure loop here and a high pressure loop here. Okay. And then we have something sort of in between with turtles, snakes, lizards. I'm spending on the tuataras we're being here too. Okay. Where we kind of have some amount of separation. They do have 3-chambered hearts, but it's not as simple as 3-chambered heart isn't a 3-chambered heart isn't a 3-chambered heart. There's some incomplete separation in the ventricle. Okay. So we get some mixing, but not nearly as much mixing. Okay. So let's look at, let's see. Okay. So let's look a little bit more closely at the birds and mammals. The question on the study guide says, provide evidence that complete ventricular separation in the hearts of mammals and in birds is homoplasious rather than homologous. Remember, this goes back to the first test. We started by talking about homology and homoplasy, right? What's the difference? If something is homologous, in both cases that it's the same, that both mammals and birds have 4-chambered hearts. But if it was homologous, what would that tell you versus if it was homoplasious?
Homoplasius is like, it was separate. Like the mammals got the 4-chambered heart this way, and then the birds got it that way.
Okay.
Good, that makes sense.
It does. It does. Dig in a little more.
Then like homologous comes from this, like the same derived trait.
So it's just a matter of learning the nerd words to really piece out what you're saying. I can tell you understand it,
Yeah.
but it's getting to be able to communicate it.
Right.
Using, okay, so here we have ventricular separation. Ventricular separation. I was going to do this. That's another way of saying 4-chambered hearts. So if it is homologous, it's this one thing that you guys learned in this class. And this is the idea of this versus this. Okay, so what is it going to look like over here? Where did the 4-chambered heart or complete ventricular separation evolve?
Down in those photos.
Down here. Homoplasius, where did it evolve? Here and here. All right. What would this organism, this ancestral, hypothetical common ancestor be? Or what would their heart condition be? Here we have a heart, 4 chambers. 4 chambers, what do you suppose this organism's heart would look like?
About 4 chambers.
Something like this. Right? Now, why do we believe this to be true with birds and mammals? Why do we believe this to be true with birds and mammals? right? Couple things. Well, look at that. I found this phylogeny on some other universities. I think it was even the Verzo webpage. I don't have an electronic copy, otherwise I'd project it here, but how do we see that on here? What's the evidence on here that if I gave you this phylogeny and I told you to describe the evidence that four-chambered heart is indeed homoplacious. I love this. This is like a whole review of the whole class on here.
Is it because like it says that it's separate, but it's not like down here where it connects them with their common ancestor? It's like past that.
Okay, so separate R and L ventricles. Separate right and left ventricles. That is on which branch?
It's on the thoracic and mammalia.
Okay, so and what would the big clade be on that? What's the largest group that includes everybody on that whole?
Synapse.
The synapses, yep. So those would be the organisms with one single temporal fenestra. Okay, so it's on the synapsid lineage. Okay, and then where else do we find it?
Archosaurian.
Yeah, on the Archosauria. Over there, that includes the crocodiles, the dinosaurs, and the birds. Okay? So that appears in those two different places. Now, who would be the common ancestor of all of those organisms? Who would that be represented by on this phylogeny? Yeah, the first amnio. So the common ancestor to birds and mammals goes way, way back to that very first organism that crawled out of the swamp and laid an amniotic egg, right? And did they have right and left ventricles separated? No. It's not real clear, because there's a lot of stuff on here, a lot of details that we're really not getting into. But certainly it would have been paired atria with one single ventricle, right? Which started all the way back at the Sarcopterygians, right? So the first vertebrate with two atria would have been a Sarcopterygian, some kind of fleshy fin fish, like a coelacanth, or all of those, the lungfishes have two atria, one ventricle, right? And that applied all the way up until the mammalians, okay? So that's a good evidence that it's homoplacious. Now, the other thing is, if we look at the mammals, if I gave somebody that's well-trained a heart, a four-chambered heart from a mammal, and a four-chambered heart from a bird, they are fundamentally different. They do have ventricular separation, but if we look, here's the mammalian four-chambered heart, and look at the way that the blood leaves the left ventricle, and it goes out basically the whole right loop of the systemic artery is retained, or no, most of the right artery is lost. Okay, we talked about how these aortic arches, about how there were 6 originally, and that they've been lost with the advent of jaws. They've been lost with the advent of gills, right? All this stuff, and then basically we keep reducing the amount of those arches. Which one of those arches was lost in mammals? That was the right, and in birds, the left systemic artery is lost. Okay, so just even fundamentally the way the circulatory system is set up, we achieve the same thing, in the birds and in the mammals, but it's clear that functionally or structurally, they are quite different. I mean, and then you can get into the actual heart, there's lots and lots of little details that are different between birds and mammals. So to answer that question, I would say, A, you can look at the phylogeny, and you can see that this ventricular separation arose independently in these two lineages, and it was not found in the common ancestor. And two, the structure of the heart, and particularly the arterial system leaving the heart, is very different in birds and in mammals. Okay? So that indicates that they're alike in some ways, but that likeness arose independently. Okay. Now, the next question, is 15. You can just cross 15 off on these study questions from chapter 12. Just cross 15 off. We're not going to get into that too much detail. Number 16, right? We're talking about flexibility. Well, really what we're talking about is these animals in the middle here. This is beneficial, right, to have complete ventricular separation. But this does have some benefits because of this incomplete separation of the ventricles. Right? There is some benefits to this. Okay, so this goes back to these other two handouts. This is a squamate, some type of lizard heart. And on the left side of the diagram, over here we see this is what blood flow through the heart looks like when the organism is air breathing. This may be something, do you guys ever see those Galapagos tortoises? They live on lava flows on the Galapagos. They dive into this cold water. They dive down and they scrape algae off of rocks until they get too cold, and then they just kind of float up to the surface, crawl out into the land and start to warm up again. Right, so they go through periods of apnea where there's not really getting a lot of oxygen. Okay. So here's when they're getting a lot of oxygen, here's the blood flow, here's when they're diving or doing something that makes them, the oxygen flow a lot less. Okay, so let's look and see. What's this and this? What are these? F2? Yep, those are the atrium. Here's the ventricle, okay? One single ventricle. So seemingly it would just mix in there like the amphibian heart, but we see that that's not the case. Okay, up here, is this oxygenated or deoxygenated blood? Deoxygenated, why?
Coming from the body.
Hmm?
Coming from the body.
Yes, it's coming from it's coming into the right atrium, okay? So over here, the left atrium is coming from the pulmonary vein, and it's oxygenated. Okay, so blood, no oxygen, comes in. Where does the oxygenated blood come in? The atrium contracts, it comes over onto this side of the ventricle. This blood is going into this little pocket down here. We'll see it's pooling in. This describes this fairly well. This thing is called venous blood enters the cavum venosum and crosses. Simultaneously, blood from the left atrium enters the cavum arteriosum, this little pocket down in here. So the oxygenated blood is coming in and filling this cavum arteriosum. When the ventricle contracts, the blood from the cavum arteriosum goes out through these two. And what are those two?
Systemic and pulmonary.
Okay, and where does systemic go?
Body.
To the body. So the blood that comes in oxygenated goes into this pouch, and it goes out through the two systemic arches to the body. That's kind of what we expect in the four-chambered heart, right? The blood that came in from the right atrium goes over here, and where does it go? It goes out this pulmonary artery. So arteries go away, and where is this going away to? Pulmonary? No, to the lungs. Okay, pulmonary. So you think about CPR, cardiopulmonary resuscitation. Okay, so pulmonary means lungs. So that, to me, this is not acting like a mixing per se, right? And why is it? It's because we have this ridge. There's a ridge in here that it's not, it kind of looks like this. We have the cavum venosum and the cavum arteriosum. Or this would be the arteriosum and the vein. So it's not total mixing. In the squamate, and it's similar for the turtle heart, is that the blood coming in that's deoxygenated stays here, and the blood that comes in that's oxygenated stays in this little separate kind of, it's not a chamber, but a little area in the heart, okay? However, if the squamate is diving, then what happens is instead of this deoxygenated blood going out to the lungs, it is pushed over and out to the body. Okay. This is the main difference. What did we that black? Oh wait, I don't have to turn it up. It's not a different slide. Here, where did the deoxygenated blood go? To the lungs. Here, where's it going? Out to the body. Why would it not go to the lungs? Because why go to the lungs? What's the point of going there? It's not beneficial, right? So we're going to bypass the lungs, and we're going to send it out to the body. That's the way that turtles and squamates do this, okay? Because they have this little ridge. It's not a total different chamber. It's not a complete separation, but it's of enough of a ridge that you can control. There's some amount of control of where the blood goes, okay? Now, so look at that. This is, I really like this diagram and the way that it describes this. Figure 12.33. This is from a different, again, a separate, different textbook. Now, if we look at it as a crocodile, billion, okay, we do have complete ventricular separation. It's indicated here, all right? So, follow the blood through, I think this is just on the back of that same diagram. Right. And I would just sit down with this and follow the blood. Comes in the right atrium. From the right atrium it goes to from the right atrium to the right ventricle, from the right ventricle to, and we can see it goes out this. What is that? It's the pulmonary artery going out to the lungs to get oxygenated. Comes back into the left atrium, right? From the left atrium down to the left ventricle, from the left ventricle out to the systemics. Okay. This is just like our heart. Okay, very similar to our heart. But the difference here, and the interesting thing that goes back to this question about the flexibility in the blood flow, okay, is right here. This little shunt, it's called the, I think it's called the foramen of Panizzi. It's a very Italian thing, very chic. Right? Air breathing, everything is cool. You got an alligator that's down under the water and it's not breathing through the air. The blood comes in, goes down to the ventricle. The blood comes in the atrium, down to the ventricle, right? But this, now the deoxygenated blood, rather than going only through the systemic arches, right? And the oxygenated blood, although it's not really oxygenated because the lungs are underwater, so they're not breathing in air, this little shunt is closed off. And so the blood from the deoxygenated right ventricle can go out through the systemic arch. Okay, so we shut this off, and rather than oxygenated blood going out to the systemic arch, the deoxygenated blood gets. It's kind of like I don't know. You shut the exit to the off of the highway, and then you just let these guys go and give them priority to go. You go instead of you, so that out to the body, we have a lot more of the blood that's deoxygenated.
Is that basically because like what you just said about it can't really go to the lungs because it's underwater, and so like you need like some type of blood through your body, so it just sends the deoxygenated blood instead?
Yeah, and in fact, that is the next question is, what are two ways in which the ability to shunt blood between the systemic and pulmonary loops would be adaptive? Okay, this is the very last step question in fact. So the question is, why has this and this evolved, right? Again, just like complete ventricular separation has evolved independently in the archosaurs and in the synapsids, this ability to shunt blood has evolved over in the lepidosaurs and in the archosaurs, so it must be adaptive. So yes, so to answer the question, if there is a little bit of oxygen in this blood, right? So it doesn't make sense for this little bit of oxygen, if it needs to get out to the systemic loop, sending it to the lungs is doing no good at all, right? It's going to make it a longer time to get to the body. If you send it to the lungs uselessly, back to the heart, and then out to the systemic loop, let's just skip that pulmonary loop because it's not doing any good, and let's just send it out to the systemic loop, okay? It will increase the efficiency in the use of whatever is left in this blood, okay? But you can think of this complete ventricular separation as a figure eight. So they're kind of like this too. At the middle is the heart. This one would be lungs, or the pulmonary loop. This one would be body, or the systemic loop. Okay? So, this is the whole point of this is to obtain oxygen, all right? And if you're underwater, you're diving, sending it over here is not going to do any good. So what we do, we basically just keep it going on this loop. Okay? That's what the foramen of Panizzi does. We don't send it out to the lungs. We're going to send it right out to the body. We're going to skip the pulmonary loop. Okay? Why? Because it's not doing anything. It's not doing anything. Allows animals to deal with periods of apnea, such as diving. Because they are not using the lungs during these periods, it is more efficient to bypass the lungs and send blood to the systemic loop. Okay? And then, so that's one reason for this shunt. And the second one, we talked about this briefly when we talked about the archosaurs, is that this blood here, the deoxygenated blood, is going to have high levels of carbon dioxide. Okay? And the carbon dioxide can be useful in the digestive system. It can be useful to produce gastric enzymes in acid. Okay? So when we send that highly carbonated blood out to the body, that includes the stomach, right? The celiac artery to the stomach. And then that carbon dioxide can be kind of harvested and used to increase the production of gastric enzymes. Okay. So not only, we tend to think of this oxygen, so there's basically this trade-off. If there's a lot of oxygen, there's not a lot of carbon dioxide, and vice versa. We tend to think of that as being a bad thing, but in that one specific case in the stomach of crocodilians, it is a benefit because that carbon dioxide is useful. Okay, so that's one reason for the shunt is, well, we're not going to lose this carbon dioxide. We're going to take it, send it out to the celiac artery of the stomach, and harvest it and use it for gastric enzyme production. Okay. So one of those, again, scientists always think about this. You get inflectable to think, well, this is because it doesn't have a lot of oxygen, it's necessarily bad. But that was one of the sort of interests. And how did they do this? They actually surgically altered little baby alligators so that this didn't work. And they found when they couldn't shunt blood that they were not able to produce as much acid in their stomach, right? So then that led them down a rabbit hole to figure out, you know, the connection between carbon dioxide and gastric enzymes. And I think that's it.
Okay. I know we talked about this, but what was there's one question on there, is that the aortic arch in the early vertebrates?
Okay.
What was that? I remember we talked about it, I can't remember.
Just what I can do?
Yeah, what it is.
Okay, yeah. So again, this would be, the way that this is kind of confusing because it's so tiny. But if we look at this one, no, I was actually thinking this morning, I'd like to get this one blown up and put on the wall in the Vert Zoo lab. But basically, what this is if you can look sideways, this would be the hypothetical first vertebrate, right? Which leads to the jawless fishes, which leads to the jawed fishes, which leads to bony fishes, which leads to lobe-fin fishes, which leads to the tetrapods to the amniotes or the amphibians, right? Main branch, amniotes, and that can split into the synapsids or the diapsids, which can become the archosaurs or the lepidosaurs, right? So if we go all the way back to that first, what would have been, we call a proto-vertebrate here. This is what their circulatory system looked like. A heart, a bunch of aortic arches, and a systemic loop. What were these aortic arches for? Why would the blood be going from the heart up to these arches? Gills. Yep, so each arch would have serviced one pair of gills. Pick up oxygen out to the body. When we go from the first vertebrate to the first jawed vertebrate, what do we see? That first arch disappears. Why? Because what happened to that first, not the actual vascular tissue, but the cartilaginous elements supporting those became the jaw. So obviously we don't need a whole loop there to pick up oxygen because it's not functioning as that anymore. We've lost that function, so we lose that first arch. Then we keep going forward, right? We have, these are fishes, so we continue to use them. right? But then when we get to the tetrapods, right, we're losing more and more, right? As we get to amnio, we've lost most of them. Why? Because we don't have any gills at all. We kept some though. Why? Because some of them need to become the carotid arteries that service the brain, and some of them still are needed to service the lungs, right? And some of them are still needed in that systemic loop. Okay? And so in that kind of, if we continue with the same story, and we look at the way that a mammal continues to lose some of those arches and retain some, and we look at the way that a bird did the same thing to accomplish the same thing, it's fundamentally different, which again suggests independent evolution of that trait, right? The hard part about this on my side is if we look at, there's so many details on here, but I don't want you to get into the nitty gritty. I want you just to be able to understand the big picture about what's going on, okay? To me, if you move forward with an understanding of this, I'm happy. We got the carotid, that's going to the head. We've got the systemic, that's going to the body. Hepatic, that's liver. Renal, that's kidneys, right? Vena cava, it's a vein. It's on the bottom. It's taking all the deoxygenated blood, bringing it back to the heart. The interior, that's the head coming back to the heart, right? We've got the pulmonary loop, systemic loop, right? Some relatively basic stuff. Okay. Well, keep in touch if you need anything. Poor Salvador had the virus and he's been quarantined twice. Ridiculous.
See you at 12:30 tomorrow.
12:30 tomorrow. Latin. Yeah. Yeah, your fish stuff coming. Okay, so you dissect our fish or something?
There was an issue with dissecting them, because some of them, really most of it, their food was already digested.
Okay,
So it was just like mush.
okay.
And there was two actually, I caught bass, I caught a bigger crappie, and the bigger crappie had 3 small minnows in it,
Okay,
and the bass already had a decomposed minnow. It
okay.
just had like a skeleton.
Oh, really? Okay. Well, then you're done with all that part.
Is that-
Yeah, so I mean, the thing is, you just want to look for anything, even if you can't identify it, just to say like,
I mean, you can
Yeah.
tell it's
I didn't do
fish,
this.
because it's like
Right.
bones and stuff.
Yeah.
But it's like.
That's okay, I didn't.
okay, but could you get the size of those items that are inside?
Dang, I knew I had to do that.
Oh my gosh.
I mean, I have a picture of them, and I can probably just, think about it before.
Okay, so what kind of data did you end up getting?
So I got the weather